In Lecture D, we get into one of the more important concepts of Chapter 17, which is blood flow, and also a very important concept with how cardiac muscle functions when we talk about the electrophysiology of that muscle tissue. So to begin, if we look at 15 steps of blood flow, we talk about later on a cardiac cycle. So we know cycles exist with different starting points, but you always come back to that individual starting point. So if you take every aspect of blood flow through the heart, through the body, and back to the heart, you would have these 15 steps. One of the best ways to get the 15 steps down and memorize those is to first know the internal anatomy of the heart. So if we do begin with the right atrium, you can see in the diagram the right atrium is there going through the tricuspid valve, then the right ventricle, up the pulmonary semilunar valve into the pulmonary trunk. The pulmonary trunk diverges then into a right and left pulmonary arteries. Those pulmonary arteries are going to let the blood go to the heart, and there from the heart to the lungs. So the pulmonary arteries are taking that deoxygenated blood from the heart to the lungs, and then there's exchange of the oxygen carbon dioxide, and then the oxygenated blood is brought back in the pulmonary veins to the left atrium. And of course you have the mitral valve, also known as bicuspid, also known as the left AV valve, into the left ventricle, up the aortic semilunar valve into the aortic arch or ascending aorta, and then to all the body cells where there's reverse exchange of oxygen carbon dioxide with the tissues, and then of course the superior and inferior vena cava bring you back to the right atrium. So again, remember the systemic and the pulmonary side of the heart. So we said that the systemic side would be the left side responsible for pumping blood out to the systems, and the pulmonary would be receiving the deoxygenated blood from the systemic area. And again, you have the various steps, but you also could tell the story of one red blood cell as it travels all the way through both the pulmonary and the systemic circuit out to the tissues and then back to the original starting place in the heart. The second concept we said we'd touch on is electrophysiology. So if you remember from Anatomy 1, the idea behind action potentials, well, we know that the heart, cardiac muscle tissue itself, is autorhythmic, meaning that it sets its own pace. It's not under conscious control because it's involuntary. So having that exception to where it doesn't need input from the central nervous system, so the CNS, the brain, the spinal cord doesn't necessarily have to send messages to the heart muscle for it to contract. So we define that as the autorhythmic property of the heart muscle itself. We also know that they're very much designed by nature for electrical movement of ions and creating electrical excited membranes that we know that will form action potentials along the membrane surface. But unlike skeletal muscles, and that's how usually it's taught in Anatomy 1, we know that most of the smooth muscles and cardiac muscles don't require the stimulus from the nervous system to generate action potentials. They automatically do it within themselves. Where we said that skeletal muscle fibers, they have to be innervated with a nerve from the CNS for that muscle to become excited and for contraction to occur. So skeletal muscle different than most smooth and all cardiac muscle. As far as the coordinated activity under electrophysiology, we know that there are certain cells that set the pace of the heart. So they're considered to be cardiac muscle cells, but because of their coordinated activity of allowing electrical impulses over the membrane, they're considered to be pacemaker cells. We know that there are other cells that will rhythmically and spontaneously generate action potentials that begin to interlock together, spread over other types of cardiac muscle cells, and of course they're designed to contract and relax. So these are known as the contractile cells. So two major cells that are found in cardiac muscle tissue, you have the pacemaker cells that set the rate, and you have the contractile cells that actually contract and relax of course, to pump the blood through the heart and then out into the other tissues. There are some very strong correlations or similarities between cardiac and skeletal muscle tissue. We know they both have the stripes or the striations, and we know that those light and dark bandings occur because of the protein fibers that are there. So just like skeletal muscle fibers have the two major contractile proteins, actin and myosin, you also find this same arrangement within cardiac muscle cells. There are some also similarities not only with the histology but physiologically how the tissues function together. So both of those have the same general function, that they generate tension within the membrane and they undergo the sliding filament mechanism. So in order to contract, we know that those actin and myosin fibers slide past one another. And typically we see that the cardiac muscle cells, instead of having many peripheral nuclei like skeletal muscle, they have one single nucleus and they branch, so again that those contractile cells can allow the entire atria to contract and then the ventricles after that. We know that they are a shorter and a much wider cell than the cardiac muscle fibers. So some similarities but some differences between cardiac and skeletal muscle. We know that because the cardiac muscle tissue is almost exclusively aerobic, meaning it needs a nutrient and an oxygen supply constantly, or else it begins, of course, to cause an infarction in the tissue, and of course we call that a myocardial infarction or a heart attack. So there's a special protein known as myoglobin. So remember, certain muscle tissue has a predominant amount of this particular protein, myo meaning muscle, a globin, a protein that's able to carry oxygen and store it in the tissue. So most of the volume of a cardiac muscle cell is going to be mitochondria that are releasing ATP under aerobic conditions. That's going to reflect the very high energy needs, the necessity to supply not only the oxygen but also nutrients to the cardiac muscle cells themselves. There are some unique structures. When you analyze cardiac muscle tissue under the microscope, we notice intercalated discs. So these literally are where the microfilaments from one cell membrane carry across and interconnect with the cell that's neighboring it, and they make this intercalated disc or this joint. So here we talk about the pacemaker cells will be joined to contractile cells and contractile cells one to another to cause the heart tissue to work like it does. We also know that in the intercalated disc we have desmosomes. Remember, that's the spot weld where literally it welds the two tissues together. And we have the gap junction or the bridge that allows ions, especially calcium for cardiac cells. The calcium has to be brought in from the ECF, the extracellular matrix, and then it has to exchange it with other cells. So this allows cardiac muscle cells to actually communicate with each other using the gap junctions. It makes it a very substantial tissue because of the intercalated disc and the desmosomes that weld the different cells together. So again, if you look at the histology, this should be very familiar. The intercalated disc, the single nucleus, the branching that occurs, and then of course the light and dark striations because of the A band and the I banding. So cardiac muscle tissue, just like skeletal muscle fibers and other cells, we know that within the membrane they have selective gated ion channels. So remember, these are specialized proteins that act like a gate to allow ions to move in or out of the cell membrane. So it's the opening and closing of these ion channels that makes both the pacemaker cells and those contractile cardiac muscle cells to create the action potentials, which we know it takes an action potential for stimulation of the entire muscle fiber to function like it should. We know that the pacemaker cells undergo a rhythmic spontaneous depolarization, so these action potentials in the pacemaker cells will spread very quickly through the heart, and they use something known as the cardiac conduction system. So the cardiac conduction system is actually a group of interconnected pacemaker cells that allow rhythmic spontaneous depolarizations within the membrane that will lead to very quick, very spontaneous action potentials. There are three different types of gated ion channels. Remember, these are specific for the ion. So notice the first one is for the sodium ion. So these are the voltage gated sodium channels that, of course, when the voltage changes across the membrane, they'll open up, and we know that sodium would influx into the cell when those gates open, or those channels. All cardiac muscle cells, except some of the pacemaker cells, have these sodium ion channels. The next one would be for the calcium ion. So the calcium ion channels are voltage gated, but they are going to be timed. So they'll open and close on a time limit. Regardless of the voltage change, there's still a timing that occurs, but later on when we talk about rhythms of the cardiac muscle tissue, of course the calcium is very important for maintaining the heartbeat. All of the other cardiac muscle cells are going to have one or more types of the channels that use or allow potassium to efflux from the cell. So we know that sodium is on the outside. A lot of the ECF is also calcium, but the potassium is located inside of the cell. So some of these are ligand gated ion gates. Others are voltage gated. So some of them respond to the actual ion, and then some of them respond to the voltage change within the membrane. So again, when you look at cardiac muscle fibers, the intercalated disc, the gap junctions, the overall makeup of having many mitochondria, that one single nucleus again, having the gap junctions to exchange those ions, and then within the membrane itself to have those different types of protein channels. Moving on with the electrophysiology, now we know that action potentials, once they begin from the pacemaker cells and then spread to the contractile cells throughout the tissue, using the intercalated discs that are going to interconnect and intertwine all of the cells together into one unit. We know again that gap junctions are going to allow for the electrical activity within the membrane to rapidly spread through all the cardiac muscle cells. So using these electrical synapses, remember a synapse is a space between a cell, but allowing ions to spread out, allowing the intercalated discs to do their job, and the gap junctions, it allows the entire heart to contract as a unit and produce that coordinated heartbeat that we consider in normal sinus rhythm. So when we talk about that you have contraction, it's because of depolarization action potentials that occur in either the atria or the ventricles. They undergo contraction or systole, and we use the term for relaxation, which is diastole. So when the repolarization happens and there is relaxation of that particular chamber, is considered to be the diastolic range. If we now focus on the contractile cells, we know that more than 99% of the actual cells that comprise the heart are contractile cells. We also know that a contractile cell results from reversing the membrane charge. So we know that the inside of the membrane is usually negative, the outside is usually positive. So it's a huge and sharp swing from about negative 85 millivolts to a more positive value of 0 or peaking at positive 20 millivolts. And this is all due to changes within those voltage gated ion channels. So those protein channels in the membrane or the sarcolemma are going to allow for unequal concentrations of especially sodium and potassium to consist on either side of that membrane. This is going to drive those ions either into the cell or out of the cell using those ion gates. So if we analyze the contractile cell and how it undergoes an action potential, we know it's a very set sequence of events that occurs. So we know that as soon as the voltage-gated sodium ion channels open up, that causes a rapid depolarization. So it was polarized, where slightly negative on the inside, slightly positive on the outside. But opening those sodium gates, we know that sodium is a very dominant ion. So those ions begin to influx or move into the sarcolemma, and is going to lead to a very rapid membrane depolarization, so a quick upshift from that -85 value. The initial repolarization phase is very small. It's a very, again, initial repolarization immediately after the spike, and this is because the sodium channels have to be inactivated, and then the potassium ions, again, they're a little bit slower to move than the sodium, so they have to move through those selective protein, or the protein channels. The potassium ions themselves are only open briefly to allow that initial repolarization. The plateau phase is very important for contractile cells. So notice with it, you have a sustained depolarization at about 0 millivolts. So this is known as the plateau phase, where that sustained depolarization happens, and this is critical because there's slow opening of the calcium ion channels. There again are more slow like the potassium, so for the calcium ions to influx into the cell, you need that plateau phase. So very important, because the calcium channels are very slow to open and you need that influx of calcium ions. Then of course the fourth phase, we have full repolarization, which is the final portion, the final step of action potentials. We know that both the sodium and the calcium channels are now going back to their resting state, and the potassium ion channels are fully open by this time. So this change in the actual sarcolemma, by closing the sodium and the calcium channels at once and leaving the potassium ion channels open, is going to allow the potassium to exit out of the cardiac muscle cell. That allows the membrane potential to go back to a resting potential of about -85 millivolts. And this of course leads to widespread repolarization within that area where the potassium ion channels are located. So your textbook gives you step by step, shows you what's going on with rapid depolarization. The sodium channels are the first to open, so notice your membrane potential in the chart. You have that quick upshift to +20. Then in step 2, that initial repolarization phase, it begins to dip slightly because the potassium channels are beginning to open. Notice that they're not fully open, but they begin to open, and potassium, I usually say in the classroom, is singing, "Should I stay or should I go now?" And it's a little bit further along to react. We said the plateau phase, which is very important, allows the calcium channels to open as the potassium channels begin to allow potassium to exit or efflux out of the cell. So there's a prolonged depolarization during the plateau phase. But then in step 4, both the sodium and the calcium channels are now closed off, and the potassium channels are fully open, so potassium is flowing out or effluxing out of the cell and causing rapid repolarization back to that resting potential of -85. So in this section we said we'd talk about two very important aspects of the heart, and number 1 would be blood flow, number 2 the electrophysiology. So make sure you start with the anatomy of the heart, the frontal section, and you're looking at all the chambers, all of the valves, all of the great vessels and their interconnectedness, and then doing the 15 steps of blood flow. With the electrophysiology, be able to define what it's like to be an autorhythmic tissue, and then understand the function of both types of cardiac muscle cells. So we said we had the pacemaker cells, but then we had over 99% of the cells which were contractile. Then be able to discuss from what you know from Anatomy 1 and what we reviewed here about the histology of cardiac muscle tissue. Pay attention to the nucleus, the striations, the intercalated disc, the gap junctions, the desmosomes. And then finally define what goes on with the types of ion channels within the sarcolemma of cardiac muscle cells. And again, we explained the 4 steps within a contractile muscle cell's action potential.